Chapter 13, RNA and Transcription. Why do you think that cells keep DNA in their nuclei? How do you think this method may be similar to processes that occur in cells? All of the parts of a cell are controlled by the information in DNA, yet DNA does not leave the nucleus. How do you think the information in DNA might get from the nucleus to the rest of the cell? And what might happen to a cell if its DNA was to be damaged? We'll be looking at all of these questions in this chapter. Before we get started, we need to look at what's called the central dogma of biology. It's kind of like the basis or the main belief in biology of how heredity takes place. This statement here, DNA codes for RNA, which guides protein synthesis, that is the dogma for biology. So we have three processes that occur: replication, transcription, and translation. So before we get into transcription, since we've already talked about replication, let's just talk about RNA for just a second. So in order to get the code from DNA, you have to have RNA. So let's compare RNA and DNA. The main differences between the two are that the sugar in RNA is ribose instead of deoxyribose. RNA is a single-stranded molecule, whereas DNA is double-stranded. And then RNA contains uracil in place of thymine. Also, DNA is a lot longer molecule than RNA, because when RNA is made, it's only the message is only taken from part of the DNA, so that's why RNA is a lot shorter. And then lastly, there are different types of DNA. So you remember we had nuclear DNA, that's the main one we always talk about. Do you remember what the other kind was? And it's this DNA you get from your mother. Now, the functions of RNA are pretty much to make proteins. It controls assembly of amino acids in proteins. And to do that, there are three types of RNA. The three types are messenger, ribosomal, and transfer. So messenger RNA is the one that actually gets the code from the DNA. And it gets it by making a complementary strand to part of the DNA, or the gene that's in the DNA. It will then take it from the nucleus. That messenger RNA travels from the nucleus to the cytoplasm, to the ribosome, and there is where the proteins can be made. Now, ribosomal RNA is made up of several the ribosome is made up of several ribosomal RNA molecules and as many as 80 different proteins. And this is where proteins are assembled. Now, when a protein is being built, you have what is called a transfer RNA, and this molecule transfers the correct amino acid to the ribosome. It is able to match up with the code that is in the messenger RNA. And when we get to translation, you'll see what I'm talking about. So, in transcription, this is when RNA is made from DNA. So, a segment of DNA, or the gene, serves as a template to produce a complementary RNA strand. In prokaryotes, RNA synthesis and protein synthesis occur in the cytoplasm. You may or may not have already known that, but since there's not a nucleus, that is kind of obvious. In eukaryotes, transcription takes place in the nucleus. Now, in order for transcription to occur, there's an enzyme known as RNA polymerase, and it is similar to DNA polymerase. It does two main things. First thing is that it separates the DNA strands. And then secondly, it is the enzyme that assembles the nucleotides into the complementary strand of RNA. Now, if you notice, when RNA is made, there's an area called a promoter region, or an area. And what that is a sequence of nucleotides that indicates to the RNA polymerase where to start adding nucleotides. So actually where the promoter region is not necessarily the area that gets coded into the messenger RNA, but it's what the polymerase is looking for to know where to start adding nucleotides. So it might be looking for, say, a guanine and an adenine in the DNA, and then from there it would start copying. And there's also a promoter area that signals when to stop making the RNA. Now, after this pre-mRNA is made, there's a little editing that needs to happen. And so sometimes some of the nucleotides need to be cut out. The ones that are cut out are called introns. The portions that are cut out are called introns, and these do not leave the nucleus. So after that little piece is cut out, the remaining pieces, known as exons, are spliced together, and that's what forms the final mature messenger RNA. This messenger RNA then leaves the nucleus and goes to the ribosome. One note about the introns and exons, and which we'll talk a little bit about later when we get to mutations, but they may possibly play a role in DNA. So for instance, if one nucleotide that is supposed to be an intron gets kept in the final molecule, that will change what protein is made. And the same thing would happen if an exon gets cut out when it's supposed to stay in the messenger RNA. That would change what protein gets made.